We will do a sheep brain dissection today. First, I will show you what supplies we need. Of course, we need the brain, and then these are the tools. We need forceps, scissor. Two things we will avoid using, that will be a sharp tip pointer. We will not use it because that damages tissue. And then also scalpel damages tissue, so we will use a long blade knife to make a neat dissection sagittally. And of course we need the blunt probe, and here I also have some pins. Sometimes those becomes handy to show the different areas within the structure. We will look at the external features first. As you can see, the brain is enclosed, encased, protected by the meninges. We can see the superior view, the inferior view. This outer layer, which we can distinctly see, this white part, that's the dura mater. When go from outside to inside, dura mater, the middle layer will be arachnoid, and hugging the brain will be pia mater. So what we will do to look at the details of the brain, we have to remove the dura mater, but first some spatial anatomical locations. So this will be frontal part. This is the posterior or dorsal. This is the inferior view of brain. Here, some important points to note. Before we remove the meninges, make sure that you take a look on the pituitary gland, the location here. We can distinctly see the optic tracts where they cross over, the specific part called the optic chiasma. So this is optic chiasma, there will be the optic tract. These are the olfactory bulbs. So once we remove the dura mater, we do not have much option other than taking the pituitary gland off. So I wanted to show you first before we remove the meninges. I am removing the meninges. Take extra care to first look how you are putting your scissor and then make the incision, otherwise you will damage the brain tissue. So I prefer to dissect following along the longitudinal fissure first. So see how I am gradually removing the meninges. This is dura mater, what means the strong mother. You can really truly see how strong is this. I am clearing the meninges on top from the brain distinctly. See, this is through this line, that's the longitudinal fissure. This is cerebrum. These are the two cerebral hemisphere, right cerebral hemisphere, left cerebral hemisphere. Posteriorly, we can see from the superior view, this is cerebellum, and here the extension, that's medulla oblongata. Longitudinal fissure separates two cerebral hemispheres, and here we can see the transverse fissure which separates the cerebrum from cerebellum and cerebrum. This is the inferior view, and here we have to be very careful. I slowly have removed the meninges so that we can save olfactory bulbs. Here I will do a very careful dissection to remove the meninges from top of the optic tract in the optic chiasma. And here going inferior posteriorly, once we remove these, we will be able to see the medulla, the pons. So I have carefully removed the dura mater from all around. This is the last part I wanted to show you. See, when we will remove the meninges completely, see, now there is no way to save the pituitary gland to keep over there. This is the inferior view, and this is the whole meninges. Let's go over through the external view with different regions of the brain. This is the frontal part. Cerebrum is divided into 4 distinct lobes: frontal lobe, parietal lobe, we are going posteriorly, occipital lobe, and on laterally are the temporal lobes. This is the cerebellum. So forebrain, hindbrain. Here, if we gently pull the hindbrain from the forebrain, we will be able to see corpora quadrigemina. So this area is the corpora quadrigemina, which is part of the midbrain, where there are 2 distinct regions: superior, inferior, superior colliculi and inferior colliculi. This is kind of remains hidden in between the forebrain and hindbrain when we are looking from the superior external view. Using a gentle force right at the middle of the brain tissue and this long blade knife, I will dissect it into 2 halves. Try not to go back and forth with the scissor. Always make a clear cut using a gentle pressure. Here, I'd like to show you a couple of other things along with the regions of the brain. See here, I have 2 different specimens. On my left hand, this is the one, the first specimen which I'm working on, and this one I dissected it before. So depending on how long the specimens are sitting in the preservative and how do we get it from the supplier, they might look a little different. On these specimen, these darker colored areas, those are mostly due to the blood vessel. Let me show you one distinct landmark in the brain. See here, there is a distinct line going across laterally. This is called central sulcus. So this region zone will be the precentral area. Here is the postcentral area. Sulcus are deep groove because there are foldings in the brain. Gyri are the ridges around. These separate the brains into different regions, and by brain mapping, we could get to know which areas are responsible for what function. And along with these, remember, sheep brain is much smaller than the human brain, much less complicated than the human brain. but of course there are similarities because these two are the mammalian system. That's why we use this specimen in the lab to show you some major areas of brain. Inside the brain, we will find 4 fluid-filled areas which are called ventricles, and those are filled up with cerebrospinal fluid. Those ventricles separate different areas of brain into forebrain, midbrain, hindbrain. So here are first 2, which are called the lateral ventricles. When we dissect, we will see it on 2 cerebral hemispheres. Around this region is the third ventricle. We cannot see it very well, but this is the area where the third ventricle, and here around the cerebellum there is the fourth ventricle, which is distinct in this section. Lateral ventricle, third ventricle, fourth ventricle. So fourth ventricle in third ventricle connected by the cerebral aqueduct. Now let's talk about different areas in the forebrain, which includes cerebrum, the largest area of brain, and then there is diencephalon, which includes thalamus, the thalamic addition, and then hypothalamus. Remains connected with hypothalamus is the pituitary gland, and in the brain there are 2 glands, pituitary gland and pineal gland. So here we see this is thalamic addition. This area is the hypothalamus. We can see the remnant of infundibulum through which the pituitary gland remains connected with hypothalamus. We can see here a little bit of the optic chiasma. This is fornix, which is at the base of the lateral ventricle, at the border of the thalamic addition. And if we track diagonally posterior from the pituitary gland, we will see the pineal gland. Pineal gland controls the circadian rhythm, and pituitary gland is in charge of a lot of different body physiology. Few smaller, relatively smaller areas, anatomically smaller areas in the brain. Mammillary bodies located at the base of the diencephalon, and then cerebral peduncle, because remember this is a different view. I have shown you the corpora quadrigemina before. That's the part of the midbrain, and then midbrain we can see this region that is called the cerebral peduncle, and then around this region is the mammillary body. I tried to keep the connection between 2 hemispheres in this section, and here I have a completely dissected view so that you can have a comparative view for 2 different segmented regions of the brain. Here, this region, which is posterior from the frontal lobe, this is called cerebellum. Cerebellum is the second largest lobe and area in the brain. So here, these are the different views for brain, and then from the posterior view, so see here, this is cerebrum, cerebellum. When we make a sagittal section, see how nicely these areas look as if somebody has painted nice design. Those are the white matter in the cerebellum and called arbor vitae of cerebellum. There are small little curves for cerebellum. Those are called folia. This is the inferior view of the brain. This part, see how all of these structures are arranged in a row. This is called the brain stem, which includes midbrain, pons, and then medulla oblongata or medulla. Up to this part, these are all the part of the brain, different parts, and this is spinal cord. Now this last part, we will look at few cranial nerves. As you know, cranial nerves originate from brain. There are 12 pairs of cranial nerves, and they are always paired because there is distribution on each side, right and left. Starting from cranial nerve 1, olfactory nerve. For sheep, we will see a very distinct structure. These are called the olfactory bulbs, which are much more prominent for sheep because for their survival, because olfactory nerve is associated with the sense of smelling. Cranial nerve 2, optic tract, and the part where they cross, this area is called optic chiasma. The third one, that also starts with O, oculomotor, cranial nerve 3. Here are those.